lab wise, we only have, I'm looking at our lab schedule, we just have 2 labs left. So we have this organic scavenger hunt lab, and we actually aren't going to sit down and do that one together. That one, when you look at the assignment, you'll just download it, and I'm going to give you until exam 4 to turn that in. So you're going to have several days to do it. But basically, the gist of it is you're looking for products, cosmetics, diet products that have any kind of a molecule that contains a functional group that we've talked about. So you just get on the internet and look up, what ingredients are in, protein supplements or cosmetic industry, or, there's so many ideas there. And you draw a picture of the molecule. I'll show you the lab. Draw a picture of the molecule and tell me what product you found it in. And so you'll have, I'll probably, what's today? Is today Wednesday? No, today's Tuesday. I'll probably give you a week to do that. Okay. So we'll have that one. So we won't actually sit and do that in lab today. We'll spend most of the day on lecture and the exam. And then next week we'll come in for the DNA isolation lab. It's a fun lab. It's really quick. And that's it for labs. The rest of the time, our lab time will be going over the material and the lecture content. So that's partly why I think we can finish by Friday. But again, we'll make that call later in the week. So any questions on the scheduling and that stuff? For now, plan for a quiz Thursday. Okay. All right. Carbohydrate metabolism. This is chapter 22. So we are kind of been jumping around chapters. But last week we did 18, 19, and 21. And now we're doing the follow-up to 21, which is chapter 22. So you guys remember in chapter 20, I believe, is carbs. We talked about carbohydrates. And that's where we introduced what a monosaccharide is, and a disaccharide, and the glycosidic bonds, and the alpha and the beta, the words like anomers. We learned how to draw them in ring structures. So now we're going to look at the main, most important carbohydrate in our body, glucose, and how that is metabolized and how that feeds into what we talked about last week, citric acid cycle and oxidative phosphorylation to make ATP. But I wanted to kind of give you this little facts on diabetes here, because this can go awry in many people, and there's devastating health kind of consequences when carbohydrate metabolism is not proper in our bodies. And the major thing that we are aware of with that is diabetes. So there's type 1 and type 2 diabetes. Do y'all know the difference between the two?
Duh. I'm just kidding.
Robin knows the difference between the two. Maybe she's well informed, you guys. Type 2 is oftentimes like exercise or diet induced. So people have very sedentary lifestyles over time of unhealthy blood glucose levels. They develop diabetes this way. Type 1 is insulin related. You have a problem with the insulin protein in your body. Okay. So that's the main difference. But I thought some of these facts were staggering. 2 of them didn't shock me. 2 of them did shock me. So like this first one right here, type 1 diabetes affects about 1.6 million people in the US. That didn't necessarily shock me. I'm aware that there's a lot of people who struggle with type 1 diabetes or who live with it, and they do well, but they just have it. And then this one didn't shock me. People who have type 1 diabetes require insulin. Okay, that didn't shock me. These other 2 kind of shocked me. Like, for example, look at this. Type 1 diabetes can be triggered by any virus, including a cold. So what this means is that we all have the gene for type 1 diabetes. We all have it. It's just a matter of, does that gene get triggered? Does it get woken up or not? And there's so much like ambiguity in the research behind this. Like, they don't know why a common cold would trigger that gene in some people, but not in others. It's really sad. We just, a family at our church, their 6-year-old little girl had to go to children. She was losing weight rapidly, having huge spikes, and just her eating patterns and all of that. The mom recognized it, and she had type 1 diabetes. And again, they don't know why. They don't know why she got it. The doctors can't say, here's why your daughter got type 1 diabetes. And so that's hard for parents, but it can be triggered by anything. And then this shocked me, but kind of what I hit on, most people with type 1 diabetes are diagnosed in the ER as children, because all of a sudden your child is having these huge crashes or extremely high blood glucose levels after a meal, and the fatigue associated with that, and just the irregularity in your body and in your blood, you end up sending your kiddo to the ER, and all of a sudden you're like, well, they had type 1. So anyways, that was pretty interesting. But I want to go back to this picture before we jump in to the content for today. So this was from chapter 21. I want to kind of use it as like a "you are here" type of map, like as if you were at a theme park. So last Friday we were down here. We were talking about citric acid cycle and oxidative phosphorylation. Today, what just happened? Where'd it go? Okay, today, sorry. It's like, I feel like I gotta touch anything, it just like weirds out sometimes. Okay, today we're right up here. We're gonna look at how we digest glucose. Well, we're really not actually gonna talk so much about digestion. We're kind of gonna get straight into glycolysis, okay? And metabolism of glucose. And notice that these things, so the end final product of glycolysis is what? It's a molecule called pyruvate. You'll see the structure of it later. And then there's some quick stuff done to pyruvate to make acetyl-CoA. And we know acetyl-CoA is the starting material for the citric acid cycle. Okay, so that's how these things are related. Remember, this is all like a sequence, a pathway, where the product of one thing is the reactant for the next thing. And that's how glycolysis ties into citric acid cycle and electron transport chain. Okay, but let's talk about glucose metabolism for just a minute, because this is not a straightforward thing. And this is kind of the beauty of biochemistry, but it's also the complex aspect of biochemistry, is that it's not straightforward. There's many different routes and pathways, and they're all interconnected. And that's kind of nice, because our body, if it doesn't need one thing, it can filter something else to do something different that it does need, and it doesn't waste anything in the process, okay? And that's kind of what you see in this whole figure here on the right. So if we have glucose in our bloodstream, there's several different things we can do with it. Now, not specifically glucose, but glucose 6-phosphate. Okay, now this is actually a reaction that was on exam 3, and we mentioned glucose 6-phosphate when we were talking about enzymes. We talked about how it's a competitive inhibitor of hexokinase, but glucose 6-phosphate is just glucose with a phosphate group on carbon 6. Okay, we could draw a picture of it. So we have the Ch2, and then it would be O, and then it would be PO3, and it would have a -2 charge. So there's the phosphate group on carbon 6, right there. And then we can finish drawing the rest of this, okay? Actually, forget the OH pattern. Doesn't matter, this first one. But glucose is right here. So down, up, down. Here we go. So there's glucose 6-phosphate. That is glucose 6-phosphate. So glucose 6-phosphate, if you're looking at the figure, several things can happen to it. It can go right, it can go left, it can go down. All right. And then there's further branch points later on when you get to acetyl-CoA. You don't have to go right into the citric acid cycle if the body doesn't need energy. So this all depends on the conditions of our body, which are changing every day. They change every time you eat a meal. It changes when you go to sleep. It changes when you exercise, changing all the time. So let's look at the different scenarios. When energy is needed, when we need ATP, what happens to glucose? What route is it going to follow? Glucose is going to be metabolized to pyruvate via glycolysis. And it's going to go into the citric acid cycle, because that is the moneymaker with ATP. That's how we make ATP. So if we need energy, and the body knows what we need. Isn't that crazy? You don't have to tell your body what to do for any of this. It just does it. We're gonna run this pathway. We're gonna run, what would be the best color? Black, maybe. We're gonna run glucose down through glycolysis. So the brown arrows, by the way, you see blue and brown. You see some green too, but the blue and the brown arrows are mainly what you see. Brown is catabolism. We're breaking things down. Okay. Blue arrows are anabolism. Okay, we're building things back up. Okay, so we're going to run glycolysis, and we're going to break down glucose to pyruvate. when we need energy. And then pyruvate will make acetyl-CoA, and that will feed into the citric acid cycle. But when we don't need energy, maybe we have plenty of ATP supply in our body. And if we still have high glucose in our blood, okay, for example, you just ate a meal, and maybe from that meal, you get all the glucose you need, all the carbs you need. And then what do? You drive by Sonic and you just swing in and you get a shake or some kind of dessert. So maybe then we have too much glucose, and we don't need all of that to go make ATP. We already have a good supply. Well, there's other things we can do with it. Okay, so when energy is not needed, glucose is going to be converted to glycogen. So this is kind of the storage molecule for glycogen, or for glucose, sorry. And the way we do that is it's called glycogenesis. Glycogenesis. So let's find the arrow. This is glycogenesis. That's this blue arrow here. Why? Because we're building up, we're linking together molecules of glucose into a long chain. So that is anabolism. All right. So that is this right here. We're going to follow glycogenesis to make glycogen. Now I'm going to pause really quick. Obviously on this chart, there's a lot of words that start with G. And I think the hardest part sometimes for me with this stuff is keeping all these words straight. Glycogenesis, glycogenolysis, gluconeogenesis, it's hard. These all where they all sound the same. So what I'm going to tell you now, the best way to keep these straight is to look at the root words. Okay, let's for example, glycogenesis. You'll see the word genesis or lysis. Okay, lysis. So think about the word genesis. Genesis is a word for creation. Okay, so glycogenesis is the creation of glycogen. Lysis, we are familiar with hydrolysis. That's a word we've used a lot that has lysis in it. That's the breakdown of something. So anytime you see a word that has lysis as the suffix, as the ending, we're breaking that molecule down. So glycogen, lysis, is the breakdown of glycogen. Okay, so that hopefully maybe helps you keep some of these words straight, because they can pile up and get confusing. All right, so that's what we can do with glycogen, with glucose. When we have enough energy in the body, we can turn it into glycogen. Or we can convert it to fatty acids way down here. We could run this in reverse and take acetyl-CoA and make lipids. All right, that's another thing we could do with it. And we know that triacylglycerols, the functional significance of triacylglycerols is that they're the body's primary energy storage molecule. So we could take glucose and turn it into fatty acids and store them that way. Okay, the last thing we can do with it, maybe when we have plenty of energy, we don't need to make ATP, we can run this pentose phosphate pathway. Okay, you remember what pentose is? Anybody? Somebody remind me, what is a pentose?
Is it a 5-carbon sugar?
Yes, okay, 5-carbon sugar. This pentose phosphate pathway, which is right up here, by the way, pentose phosphate pathway, converts glucose to NADPH. What is NADPH used for? What is NADPH? Anybody remember? It's the cousin to NADH. It's just like it. It's a reducing agent, okay? And we use it to go to the citric or the electron transport chain, and we oxidize that, and that feeds the entry of electrons in the electron transport chain. Okay, so it's a reducing agent. And we don't need it just for the electron transport chain. There's plenty of biochemical reactions throughout the body that depend on redox. Okay, so we need a constant supply of oxidizing and reducing agents in the body, all over the body. And NADPH is useful for that. Okay, so we can make that, or we can make ribose 5-phosphate, which is a precursor for nucleic acids. All right, so glucose can even be used to make DNA or RNA or things that we need. Okay. So there's several things we can do with glucose, all right? Just depending on what conditions are present in the body. Now, if we need glucose, and let's say we're not getting enough of it in our diet, then we can also synthesize glucose from non-carbohydrate sources, all right? in a process called, this is the hard word, gluconeogenesis. It's that one right there, the blue arrow. So genesis, the creation of glucose. So gluconeogenesis, all right? So that's where we can take lactate, pyruvate, and we can recycle and make more glucose if needed, okay? All right, you also see, I want to point out that amino acids and then glycerol from lipids also can feed into gluconeogenesis. So what that means is that if our body doesn't have enough glucose, glucose is the primary preferred fuel for the brain. Brain cells have to have glucose, okay? But if our body just doesn't have enough of it, we can take other things, other biological molecules, and we can make glucose if we're not feeding ourselves enough of it. And that's kind of a handy thing for the body to be able to do. All right, so let's talk about glycolysis. Glycolysis is the pathway for glucose metabolism. All right, we call it glycolysis. So it's the biochemical pathway that breaks down glucose into what? 2 molecules of pyruvate. And then we also get 2 NADH, and we get 2 ATP. Okay, so these are the overall products of glycolysis. Now, pyruvate will just go on into make acetyl-CoA in the citric acid cycle, and we can get more ATP there, but we do get an overall net of 2 ATP directly from glycolysis and then 2 NADH, right? Overall, glycolysis is broken down into 2 parts. There's 10 steps in glycolysis. We're going to look at all 10. The first 5 steps, steps 1 through 5, we call the energy investment steps. So we have, that's where there's 2 different steps there that we have to use ATP. And we have to invest a molecule of ATP. Steps 6 through 10 are known as the energy generation steps. This is where we're going to get back some ATP. And overall, we'll get a net of 2. Okay. So I'm gonna read this last bullet point in just a second. So I want you to look, this is kind of an overview. Here's glucose, and if we throw in 2 ATP, these arrows represent steps 1 through 5. Whoops. Now, the one thing that's a little deceiving about the graph and the way I've kind of made this figure is that we don't run steps 1 through 5 twice. So I don't want you to think this happens twice, but we do end up with 2 molecules of this glyceraldehyde 3-phosphate, okay? We do end up with 2 of those. We don't run steps 1 through 5 twice, but we do end up with these 2 molecules, and you'll see how in just a second, okay? Now, what's not deceiving about this graph is the fact that we do run steps 6 through 10 twice for each molecule of glyceraldehyde 3-phosphate. Okay, so honestly, it would probably be more accurate if I had just a single arrow here and then it broke it off at the end like that, because we only run steps 1 through 5 once for each molecule of glucose. But steps 6 through 10, since we get 2 identical products, we run the back half twice, okay? And then we end up with 2 molecules of pyruvate from that. So that's just kind of and each time you run step 6 through 10, you get 2 ATP. So can you see how we yield a net overall of 2 ATP? We have to put in 2 ATP here, but then for twice we get 2 back. So it's like we get 4 on the product side, and we've invested 2, so that's an overall net gain or production of 2 ATP. Questions on that? Is that confusing to anybody?
Wait, so on the product side, sorry, I think I'm just going to restate what you just said, but on the product side, we get 4 total.
Yes.
Is that what you're saying? Okay, yeah.
2 ATP for each molecule of glyceraldehyde 3-phosphate that's converted to pyruvate.
Okay.
So we produce 4 molecules. It's just that we had to invest 2 to begin with.
Mm-hmm.
So we get a net yield of 2.
Okay.
It would be like thinking, if this helps you, you could say, oh, well, these 2 cancel out that 2, so we get a net yield of 2. Okay.
Mm-hmm.
If that helps you see it that way. But we invest 2, we produce 4, so we yield a net of 2.
That makes sense.
Okay, here's the actual picture of all of glycolysis and its intermediates. And honestly, this is not as clear as I would love it to be. So I'm afraid if I zoom in, it's going to get blurry, but I'm going to zoom in some. So we're going to start up in the top left-hand corner. Now, what do I want you to know from glycolysis? I'm not going to have you draw everything out, but I do want you to know for this particular pathway, I want you to know which steps require ATP and which steps produce ATP, okay? And then I want you to specifically know the enzymes in the first 5 steps, okay? I want you to know the enzymes for the first 5 steps. We're going to talk about which enzymes are regulated and how they're regulated. And then I want you to know where ATP is involved in this process, okay? And especially for the first 5 steps, you need to know the intermediates, like the names of the intermediates. So the glucose 6-phosphate, the fructose 6-phosphate, fructose 1,6-bisphosphate, those things. And then you obviously need to know the name of the final product, pyruvate. So let's walk through it, and I'll point out things as we go. And if you have any questions when we get done of what you do need to know for sure, we can follow up then as well. So let's start with glucose. This is blurry. So this is the enzyme. I'm actually tempted to just write over this. This is the enzyme hexokinase. I'm going to rewrite this. Maybe you can read it on your paper. I don't know. Hexokinase. And this is the enzyme that runs the first step in glycolysis. Now, when you run this first step in glycolysis, you're not actually committing yourself to glycolysis just yet. Like, even though yes, this is technically the first step, you're not committed to the pathway of glycolysis until later on. Because remember, with glucose 6-phosphate, which is the first product in this chain of 10 steps, there's several things that can happen. Glucose 6-phosphate doesn't have to go through all the way through glycolysis. That doesn't mean you have to go through glycolysis. You could run the pentose phosphate pathway, glycogenesis. You could go different ways, all right? So it is technically, we say, the first step in glycolysis, but there's still other things could be done here. So we're not committed just yet, but this does require an ATP. And what's happening is that we are phosphorylating this carbon 6 right here. And I'll highlight it that this is the phosphate group now on carbon 6. Everything else is the same. Okay, nothing else changes structurally. So hexokinase is there. And I don't know if you guys remember, this was on exam 3, that, I'm gonna try to use a pink arrow for this. Glucose 6-phosphate, this is actually, I'm gonna draw it like this and put a little arrow. That's kind of a common way to represent an inhibitor, okay? It can inhibit hexokinase. So if we don't need to make a lot of pyruvate, if we don't need to make ATP in the body, if we have plenty of those things, glucose 6-phosphate can inhibit hexokinase and say, no further, we don't need to make anything else, let me go do something else, all right? We can use glucose for other things in the body. So it's an inhibitor of hexokinase. So that's one way the pathway is regulated right there at step 1. Now, there's been a lot of different research. There's more than one method, I believe, that glucose 6-phosphate can inhibit hexokinase. If you read in chapter 19, it tells you, it's a competitive inhibitor. In fact, I use this as an example of competitive inhibition. But if you read in chapter 22, it'll tell you it's an allosteric inhibitor. So it's perhaps that glucose 6-phosphate can inhibit allosterically at a site different from the active site, or competitively in the active site, because glucose 6-phosphate does look a whole lot like glucose. So it would make sense that it could fit right there in the active site. All right, so that's one type of regulation right with step 1. Let's go on to step 2. Step 2 is just an isomerization. So we take glucose 6-phosphate and we just rearrange it to fructose 6-phosphate. So think about glucose and fructose. The difference there is that you have a 6-member ring. Fructose is a 5-member ring. Okay, you see how it's a 5-member ring in fructose? So all we did is we took this whole, I don't know if this is going to help. Let me highlight it in red here. We took this whole carbon group with the OH and we just, now it's oriented up. All right, and the ring closure is smaller. and it's a 5-member ring. Now that's fructose. We still have the phosphate group on carbon 6, but we just kind of cinched the ring a little bit, turned it into fructose. So that's just an isomerization. The next step requires another investment of ATP. Okay, so steps 1, by the way, these little, you probably noticed this, these little numbers up here tell you the steps, step 1, 2, now we're on step 3. So step 1 and 3 require an investment of ATP. Step 1 and 3. And this is catalyzed by phosphofructokinase. I'm going to rewrite that. Phosphofructokinase, in case you can't read it on my screen. Remember, the word kinase is a kind of enzyme that catalyzes specifically phosphorylation. Okay, kinase. So we're just going to phosphorylate carbon 1, that carbon that we just bent up in step 2 and is now sticking up above the ring. Now we're going to phosphorylate it. So now we have the original phosphate group on carbon 6, plus we have this new phosphate group on carbon 1. And now we're done with the energy investment steps. We have one last thing we have to do. And well, we're almost done. 4 and 5 technically are classified as energy investment. One more thing we have to do before we have this intermediate that is then ready to generate some ATP. But let me talk one more, let me share one more thing about step 3. Step 3 is another really critical regulatory step in glycolysis. So I'm going to call step 3 okay, this is the most critical one, actually, more critical than step 1, because there's a lot of things that can inhibit or activate phosphofructokinase, okay? So I want you to think about this for a second. ATP in the body, the way we use ATP, I'll draw a picture of this right up above, is we take ATP and we hydrolyze it, okay? We turn it into ADP, or in some cases, you might even see AMP. Anybody know what AMP would stand for? So ATP is adenosine triphosphate.
Is it mono?
Yeah, monophosphate. So we hydrolyze either one phosphate group off of ATP and that releases 7.3 kcal of energy, or we can actually hydrolyze 2 groups off of ATP and we end up with adenosine monophosphate. That releases energy as well. And as we use ATP in the body, these things build up, right? As ATP is used. And that would be a really heavy indicator to our body. Hey, our supply of ATP is getting low. We need to make more, right? Because we're using it. So ADP and AMP are actually activators. They're allosteric regulators. But if you remember when we talked about allosteric inhibition, I just feel like I want to pull that slide back up. We talked about allosteric inhibition. We said there's activators and there's inhibitors. So I'm going to go down the picture that I'm thinking of in my head, right? Okay, right here. So AMP are positive allosteric regulators. So this would be like AMP or ADP. And this would be phosphofructokinase. I'm going to abbreviate. And they can come in and they can bind and say, hey, we really need to run glycolysis. Like, come on, let's make more ATP, all right? So they definitely activate the pathway here. But we also have some negative allosteric regulators, okay? ATP would be a negative allosteric regulator for phosphofructokinase. Phosphofructokinase. Okay, ATP, if we have a good supply of it, it can come in and say, hey, we really don't need to run glycolysis right now. Y'all go do something else with glucose, okay? Don't run glycolysis, we've got plenty of ATP, all right? So this is a major regulatory point in glycolysis, okay? This pathway is very temperamental, all right? And there's a lot of control points because of what it does for us in our body. Okay, so questions on that? Step 3 is a really important step. I definitely want you to know what are positive allosteric regulators for phosphofructokinase? What are negative allosteric regulators for phosphor? Citrate's another one. I forgot to write that down. Citrate. ATP and citrate are both negative allosteric regulators. So know these 4 things and how that applies to glycolysis. Let's go back to our glycolysis. So that's step 3. And at the end of step 3, we now have a product known as fructose 1,6 bisphosphate. That's just acknowledging there's a phosphate group on carbons 1 and 6 now. Okay, questions so far on the energy investment steps?
You said citric what?
Citrate. It's like the anion form of citric acid. Citrate.
Is the number for the free energy, is that how many ATPs being used?
So the -4 and the positive 0.4, those numbers?
Yes.
I was actually going to talk about those, Madison. So you notice that the steps that involve ATP have a negative delta G overall, meaning ATP, these are coupled reactions, okay? Coupled reactions, and this allows the process to, ATP, the hydrolysis of ATP supplies the energy for this reaction to happen. And you just get a little extra energy released that's probably released as like body heat, okay? It helps maintain body temperature or something. But this doesn't tell you the number of ATPs needed. It just tells you how much energy is released when this reaction is run. So look at the difference between steps 1 and 2. Step 1, it releases 4 kcal of energy per mole of glucose that's phosphorylated. But step 2, it has a positive delta G. This is actually not technically a favorable reaction. Now it's barely positive, 0.4. Okay, but that's why we have the enzyme to help us run and catalyze this reaction. Because on its own, it's actually an endergonic reaction, not exergonic. But step 3 on its own probably wouldn't happen, but coupled to ATP, the hydrolysis of ATP releases 7.3 kcal of energy, and that's used to run this reaction. We get an excess left over of -3.4 energy released, okay? So it doesn't tell you how many ATP are needed, it just tells you how much energy, excess energy is released when you couple the hydrolysis of ATP to these reactions in glycolysis. Does that make sense? Okay. All right, let's keep going. So now steps 4 and 5. So we're now going to take fructose 1,6 bisphosphate, go back to red, and we're going to cleave it into 2 products. So we're going to like cut it down the middle, okay? So we're going to cleave this, and this won't be a perfectly straight line, but we're going to like cleave this in half basically. And we're going to end up with 2 slightly different products. One is known as dihydroxyacetone phosphate. Sometimes they call it whoops, that's not a D. DHAP. That's the abbreviation for that. Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, which is the one we're more interested in. Glyceraldehyde 3-phosphate feeds on into the next cycle, okay? Dihydroxyacetone phosphate is simply converted to glyceraldehyde 3-phosphate, okay? So you get this aldolase enzyme that runs this reaction, and it splits the fructose 1,6 bisphosphate into 2 products. And then you have triose phosphate isomerase is the enzyme that simply takes DHAP and converts it to glyceraldehyde 3-phosphate, okay? If you look at these 2 molecules, they're really not that different. They both have a phosphate group, different places. They both have a carbon double bonded to oxygen, and then they both have an OH group on a carbon. They're pretty similar. It's just they're rearranged differently. So we just take DHAP and we convert it into, we isomerize it into glyceraldehyde 3-phosphate. So we end up with 2 molecules of glyceraldehyde 3-phosphate when we cleave fructose 1,6 bisphosphate. We end up with 2 molecules of glyceraldehyde 3-phosphate when we cleave fructose 1,6 bisphosphate. So I'm going to write that down. We get 2 of these. All right. I definitely want you to know which step is the cleavage step. And what's the product of the cleavage step? So I want you to know this word, glyceraldehyde 3-phosphate. That is the product of the, ultimately you get 2 of these when you cleave fructose 1,6-bisphosphate. Okay, now we're done with the first half of glycolysis. That's steps 1 through 5. Now we have the energy generation steps. So I'll move through these a little bit faster than I did those first 5, just because I'm not going to address the regulation, or most of the regulation that's important happens in those first 5 steps. But I will walk through where we get the ATP. So glyceraldehyde 3-phosphate is going to be converted into 1,3-bisphosphoglycerate. Okay, so we're going to phosphorylate this other carbon up here. Okay, so now we have 2 phosphate groups, 1,3-bisphosphoglycerate. And glycerate sounds a whole lot like glycerol, and you guys should be familiar with the structure of glycerol, remember? So that might actually help you remember structurally what these molecules look like. So we have 2 phosphate groups, 1,3-bisphosphoglycerate. We do have to use a molecule of NAD+. Whoops, I'm on highlighter. NAD+. If you guys remember, NAD+ was used a whole lot in the citric acid cycle. This is an oxidizing agent. So we reduce NAD to NADH, and in the process, we phosphorylate the glyceraldehyde 3-phosphate. Now we have 1,3-bisphosphoglycerate. Then we are going to phosphorylate and make ATP. So we're saying that we're using a phospho. And I don't know if y'all can read the names of these enzymes very well. Can y'all read the names of the enzymes?
No, not really.
I'll try to find a better figure. I want to write this one out because this is a kinase, okay? And remember, kinase, those enzymes, they catalyze phosphorylation. So what are we phosphorylating here? We are going to phosphorylate ADP and make a molecule of ATP. Okay, this is our first generation of ATP on step 7. You can see the 7 there. This is step 7. We get our first ATP back. Now remember, this is done twice because we're running 2 molecules of glyceraldehyde 3-phosphate through this cycle, okay? So we'll get an ATP on step 7 for each of these, each glyceraldehyde 3-phosphate that's run through these steps, okay? Step 8, the leftover product is known as 3-phosphoglycerate. You don't have to write these down. And then to step 8, we simply kind of rearrange. We make another isomer to 3-phosphoglycerate, make 2-phosphoglycerate. So all we're doing, this is a phosphate group right here, and it's just being moved up to the next carbon. So we're just rearranging the molecule. That's step 8. Nothing really significant there. Step 9, we lose a water molecule. So we cleave off a water molecule. Carbon 2 and 3 are involved. We're losing this OH group from carbon 3. We don't have that on this phosphoenolpyruvate down here. So we lose this water molecule, and now we have phosphoenolpyruvate. The last step is the conversion to pyruvate. So this molecule, phosphoenolpyruvate, is the next to last product. And that is converted to pyruvate. Sorry, this keeps moving. And in the conversion from phosphoenolpyruvate to pyruvate, we're going to again make ATP. So we're going to take this phosphate group. Y'all can see this ADP molecule is converted to ATP here. And that's the second generation of ATP. So step 7 and step 10, we get an ATP back. Our final product is pyruvate. Ultimately, remember, we get 2 of these. Well, I'll just underline it. We get 2 of these because we have 2 glyceraldehyde 3-phosphate that run through these steps, okay? So the most important steps here are steps 1 through 5. Know the enzymes. I'm going to try to find, like I said, a clearer figure. I didn't realize how grainy this would be when you zoom in. Know the enzymes that catalyze steps 1 through 5. Know which steps on the energy generation steps actually give us an ATP. Know that pyruvate is your final product and you get 2 of those. Step 6 is where NADH is produced. NADH or NAD plus is reduced to NADH. And then I want you to know which enzymes are important regarding the regulation of glycolysis. So those enzymes I wrote out: hexokinase and phosphofructokinase. And this is feedback inhibition, right? We're taking products later on in the chain and we're talking back to the enzymes that way. And that's feedback inhibition, where a product later in a pathway or a sequence comes back and inhibits the enzyme that catalyzes a step earlier in the reaction.
Hey, Dr. Everly, could you repeat the last thing you said we need to know from this?
What was the last thing I said? So let me go through the list again. Know the enzymes, the names of the enzymes that catalyze steps 1 through 5. Know the names of the intermediates, because on the exam I might say fructose 6-phosphate is catalyzed to fructose 1,6-bisphosphate. Okay, so for sure, for steps all the way up to glyceraldehyde 3-phosphate. I'll highlight in pink the names that I want. This name: glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, the dihydroxyacetone, glyceraldehyde 3-phosphate. Know the names of these intermediates, and I want you to know this product down here, pyruvate. Know the names of those. I'm not going to ask you to draw them out, but if I showed you a picture, or if in a word problem I said, you know, pyruvate or glyceraldehyde 3-phosphate, I would want you to understand what part of glycolysis that's relating to. Okay, know the inhibition with phosphofructokinase and hexokinase. What things inhibit those or regulate those in a positive or negative way. Which steps require the investment of ATP, and then which steps produce ATP. So steps 1 and steps 3, we have to invest ATP. Steps 7 and steps 10, we produce ATP. And then step 6 involves the reduction of NAD plus to NADH. Okay, questions on that? This is a beast of a pathway. All right. Now, once we make pyruvate, so let me see how far we hopefully gonna get today. I'm gonna go 1, 2, 3, 4. I think we're gonna, yeah, we'll probably just get to here, regulation of glucose, this slide right here, and then we'll take a break. So 4 more slides and let's take a break. All right, metabolism of pyruvate. So there's a couple different things that can happen with pyruvate after we make it from glycolysis. All right, so if you remember from the figure up top, pyruvate can be metabolized further to acetyl-CoA. And it could also be turned into lactate depending on what the body needs, if it needs ATP or not, and depending on if we have an aerobic or an anaerobic system. You guys know the difference between anaerobic and aerobic? What's the difference?
One requires oxygen and then the other one doesn't. Like
That's right.
anaerobic doesn't require oxygen.
Right. Aerobic sounds like air to me. So I think, okay, we got oxygen in aerobic systems. Anaerobic, no oxygen, okay? All right. So let's look at aerobic oxidation of pyruvate. So pyruvate under aerobic oxidation, and that's following the downward arrow. This is technically aerobic oxidation of pyruvate is what's going to give us acetyl-CoA. Right? So aerobic oxidation of pyruvate, it moves into the cytosol of the mitochondria, because remember, in the mitochondria is where the citric acid cycle takes place. Glycolysis, I didn't mention this earlier. Sorry, I meant to mention this. Glycolysis happens in the cytosol of your cells. So outside of your matrix or your mitochondria, just in the cytosol, that's where glycolysis happens. And so if we're gonna continue towards citric acid cycle, oxidative phosphorylation, now pyruvate is gonna move into the mitochondria. Okay, so it's gonna move from the cytosol of your cells into the mitochondrial matrix across both the outer and the inner membranes. And in the matrix, that is where it's called pyruvate dehydrogenase. It's going to convert pyruvate to acetyl-CoA, and this is what the reaction looks like. So here's pyruvate. You've got this 3-carbon molecule. Here's just kind of the abbreviated form of coenzyme A. And basically, this enzyme, a dehydrogenase, it's going to add the acetyl group. So we're just going to add this part of pyruvate. This is an acetyl group. We're going to add that to coenzyme A, and these will be formed together. That's acetyl-CoA. Now, in the process of this, we're going to reduce NAD plus to NADH. Okay. And we do get a molecule of CO2 from that as well. Okay, so that's one fate of pyruvate. It can just be turned into acetyl-CoA. That's under aerobic conditions. But what happens in certain tissues or cells or organisms that don't have a readily available supply of oxygen? So red blood cells do not contain mitochondria, and muscle tissues contain a low supply of oxygen. And both of these things, we know oxygen is essential for electron transport. It's the final electron acceptor, right? That's you make those molecules of water. You've got to have oxygen there to do that. So if we don't have supply of oxygen and we don't even have mitochondria, then we cannot run electron transport. So how do we generate energy for our muscle tissue? And how do we generate energy for our red blood cells? These are cells and they need energy. How do we do that if we don't have oxygen and we can't run electron transport? Well, there's another pathway. You know glycolysis generates ATP, right? Not a lot, but some, okay? I think from 1 molecule of acetyl-CoA, did anybody figure out how much ATP? This was a question on the exam. How much ATP can you get from 1 molecule of acetyl-CoA? Y'all remember? The answer should have been 10 ATP, okay? 10 ATP. So quite a few ATP from electron transport and all of that. So we don't get that many ATP from glycolysis, but we still make ATP from glycolysis. So as long as we can run glycolysis, we should be okay. We just have to run it continuously. And that is what can happen under anaerobic conditions. So instead of converting pyruvate to acetyl-CoA, in anaerobic conditions, we'll convert it to lactate. So we're going to take NADH, we're going to rip the hydrogens back off of NADH, we're oxidizing NADH, and we're reducing pyruvate. Okay, we're reducing it to lactate. And then in highlighted, you see there the carbon that did have a double bond to oxygen now has an OH group. So pyruvate to acetyl-CoA, this is considered an oxidation reaction. We're reducing the coenzyme, but we're oxidizing our substrates. But in anaerobic conditions, we're reducing pyruvate and we're reoxidizing our coenzymes. It's the opposite. And in case you just need more of a visual, sometimes I like to just throw these visuals on. This is a graph not in your textbook, but essentially it's kind of showing you what can happen in anaerobic conditions. So glycolysis makes ATP. We know that, 2 ATP. And if we don't have oxygen available, pyruvate can be reduced to lactate, okay? So we'll get 2 pyruvate every time we run glycolysis. So we'll make 2 lactate. And in the process of doing that, we take NADH, right? And we're reforming our NAD+. NAD+ is needed to run step 6 of glycolysis. If you go look at the figure up above, step 6 needs NAD+. So this is kind of a self-feeding cycle. We're regenerating NAD+, we can keep running glycolysis, we can keep generating. This is how we're going to produce ATP. And this is how we fuel cells like our muscle tissue and our red blood cells in the absence of oxygen. And it's not that oxygen is needed for any of these steps right here. We don't need oxygen. Is not needed here. So why are we calling this anaerobic? We don't need oxygen here. But where do we need oxygen? Where do we need oxygen? It's later on. Where do we need oxygen later on? In what? Electron transport, right? It's the final electron scepter and electron transport. So it's not that we need oxygen here, but we don't want to go do a whole bunch of stuff and run a whole bunch of reactions and use a lot of energy trying to get to electron transport if we can't even run electron transport in our red blood cells or in our muscle tissue because they don't have mitochondria and we don't have an oxygen supply. Does that make sense? We can just generate our ATP this way from glycolysis. There is another little section in your textbook that talks about the fermentation of pyruvate. This is done like literally the beer making process with yeast. It'll take pyruvate and ferment it, okay? But don't skip, I'm not gonna ask you questions on fermentation. So if you're reading through the textbook section as well, just skip over the fermentation part. Okay, so now let's look at, we've talked about glycolysis right here. And we already know how much ATP we get from the citric acid cycle and electron transport. So let's figure out how much energy are we generating total from a molecule of glucose. If we run a molecule of glucose all the way through glycolysis, make acetyl-CoA through the citric acid cycle and through electron transport and oxidative phosphorylation, how much energy do we get all together? So I've summarized right here on each of these steps or stages what the overall energy products are, okay? The NADH, we know NADH is useful for making ATP. That's what feeds electron transport. FADH as well. FADH2, I should say. And then how much ATP we get, the small amounts we get from glycolysis and citric acid cycle along the way. So now we need to add all of this up. So how many molecules, the question is how many molecules of ATP are produced from one molecule of glucose in the body? So the first things we need to do, number one, determine the total number of NADH. So look at our three things here. We get 2 NADH plus from glycolysis, 2 more from the production of acetyl-CoA. Why do we get 2 from the production of acetyl-CoA? Right here. Remember, this happens twice. We produce 2 pyruvate at the end of glycolysis. So this happens, we could say, times 2. Because both of those pyruvates would be converted to acetyl-CoA if we're running aerobic oxidation. Or under aerobic conditions, I should say. So that's why we have 2 NADH right here. And then altogether from the citric acid cycle, which is going to be run twice, we have to double everything because we get 2 molecules of acetyl-CoA. So we're accounting for everything produced from 2 molecules of acetyl-CoA. 1 molecule of acetyl-CoA made 3 NADH and 1 FADH2. 2 molecules of acetyl-CoA make 6 NADH, 2 FADH2, and 2 ATP. So total, we get 10 NADH, if you added that up right. I think I'm right. Double check me. And 2 FADH2. So anybody lost? Who's lost? I think the hardest part is remembering, like, doubling everything because you get 2 pyruvate from glycolysis. And so then you just got to double everything from there. I think that's the hardest part. Okay, now remember, each NADH that enters the electron transport chain, I rounded up here. Technically, it's 2.5. Maybe I should be a little more accurate. You know what? I will. I'm going to cross this out. I'm not going to round up. 2.5. 2.5 ATP and FADH generates 2 ATP from the electron transport chain, okay? So remember these steps. Now we're gonna say, add it all up. How many ATP would we get? So if we have 10 NADH, we know that for every, we know that 1 NADH makes 2.5 ATP. So we can do, my writing is really spot on today. 2.5 ATP, so 10 times 2.5. This is how much ATP would be produced just from 10 molecules of NADH. So 25, right? And then 2 FADH2 times for every 1 FADH, it's 2 ATP. So that's 4. This is ATP, by the way, ATP. Plus, how many ATP do we get from glycolysis? There's 2 there. And from the citric acid cycle, be 2 there. So we need to say this plus 4 more ATP. 2 from glycolysis, 2 from the citric acid cycle. How much do we get total?
So you just add everything right there.
Yeah, 25 and 4 and 4.
33.
33. Now, if you read in your textbook or on the internet, you might see 38. And that's if you use the relationship that each NADH makes 3 ATP. I crossed it out and put 2.5 because that's more accurate. Okay, but if you're using the relationship of 3, you might see 38. So don't think you're wrong. We're not wrong. And these systems are not perfect. They're faulty too, okay? The bilayer in the mitochondria is leaky dihydrogen ions, and so not everything always goes the way it should. So in a perfect system, it might be 38 ATP. In a real world system, it's definitely going to be lower, okay? All right. How many slides? I'm on slide 7 of 16. I might stop here. I think this is a good stopping point, actually. And then we'll move on. We're gonna take, let's see. I probably need 30 minutes to take pictures of your exams and email them to you guys. I'll get all that ready to go. Let's get back on at 11 o'clock. Let's finish glycolysis, finish talking about the remainder of the slides we have left. And then we'll go over exam 3. Okay, we're not actually gonna be doing a lab activity today. I will probably download that PDF and just walk you through again what you're supposed to do on that. But that's not going to be due today, so we're not going to spend time together. That'll be more of an independent activity. So, any questions on what we've covered this morning? All right, well, I will see y'all at 11 o'clock.